Sixth-year biology students sitting here leaving cert in 2026, we've already covered DNA, deoxyribonucleic acid. We've talked about that it's found in the nucleus, that it's double-stranded helix, meaning that it can't actually leave the nucleus because it's too big. But what we didn't talk about is that DNA is made up of sections known as genes, and different genes contain different codes to make protein. But we know that proteins are made outside of the nucleus in the organelle called the ribosomes. DNA, however, can't leave the nucleus. So how are we getting the codes from those genes that are found on DNA to the ribosome? What happens is DNA sequence makes a copy of itself onto a single-stranded molecule called RNA. And it's a particular type of RNA known as mRNA. So we're going to talk about what RNA is before we get into the three different types. So RNA is single-stranded. It stands for ribonucleic acid. Its basic unit is known as a nucleotide because RNA is a polymer made up of smaller monomers, and the monomer is a nucleotide. The monomer of RNA is composed of three different parts. You have a phosphate, a 5-carbon sugar called ribose, and you have a nitrogenous base. Nitrogenous bases are just made of carbon and nitrogen. RNA can have adenine, guanine, cytosine, or uracil. So remember, in DNA we had thymine. We have no thymine in RNA, we have uracil instead. So when we're doing our complementary base pairs, adenine bonds to uracil, guanine still bonds to cytosine, but A bonds with U instead. So the differences so far: RNA is single-stranded, DNA is double-stranded. RNA has uracil, DNA has thymine. RNA is shorter, DNA is longer. The sugar in RNA is ribose, the sugar in DNA is deoxyribose. So where do we find this RNA? We find it in the nucleus because it's going to carry the code to make the protein out through the nuclear pore, into the cytoplasm and into the ribosome, which means we also find it in the ribosome. There are three different types of RNA that we need to know. We have mRNA, which stands for messenger RNA, and we'll do more on this in protein synthesis. And that's the mRNA that's going to be made in the nucleus to carry the code out of the nucleus into the ribosome. And it has different groups of bases, again, like we covered in our DNA, called codons. Those codons, each 3 bases, will code for a particular amino acid. And the particular sequence of amino acids determines the particular protein that is made. So mRNA, messenger RNA. It has the sugar phosphate backbone, and it has your bases adenine, uracil, guanine and cytosine, and they are grouped into groups of 3 called codons. Each codon is going to code for a particular amino acid, and make sure you know how to get the complementary base pairs. So for example, if we had CUC, AGU exposed, the complementary RNA sequence would be GAG, UC. So make sure you know the complementary base pairs, okay? And remember, those complementary base pairs are bonded together by hydrogen bonds. So what we need to know is that the mRNA is going to be coming from the nucleus into the ribosome, and it has bases that are in groups of 3 known as codons. A sequence of 3 bases known as a start codon will cause protein production to start. The sequence of codons in the middle will cause for a coding of a particular amino acid. And then a sequence of 3 bases, the stop codon at the end, will cause protein production to stop. Another type of RNA we need to know is ribosomal RNA, and that's what the ribosome is made of. And the third type of RNA is tRNA, transfer RNA. That's going to transfer in an amino acid into the ribosome that the mRNA has determined which one is going to come in. But I'll talk more about that in protein synthesis. For now, make sure you know ribonucleic acid is single-stranded. It doesn't have thymine, it has uracil. It has the sugar ribose. It is a polymer made up of basic units known as nucleotides. And make sure you know A bonds with U and G bonds with C, because you could be asked, you could be given a complementary sequence for DNA and asked to write the mRNA sequence, or you could just be asked to write a complementary mRNA sequence. So make sure you know how to do all of the following, and in my protein synthesis video I will do an example of the complementary sequences.